Hi to all of you and welcome back to the series of molecular biology lectures. This is the 15th lecture of the series, and in this lecture we will be discussing the translation process, its aspects and the various phases of translation. As we are aware that central dogma of unidirectional flow of information involves the three important processes which are central to it: replication, transcription and translation. Translation, like other two processes, also involves three phases: initiation, elongation, and termination. Initiation phase is the phase or the start of the translation takes place. In this phase, where the components of different translational machinery, particularly the ribosomes, mRNA, and the tRNA, together with the amino acid, come together to form an initiation complex. So it involves the interaction of an mRNA first with the small subunit of the ribosome, which I have already told you in the previous lecture. The interaction happens between the Shine-Dogano sequence with the 16S sequence of the small subunit, so as to direct it right ahead of the start codon. This interaction involves the initiation process of the translation. Before we begin with the actual initiation, we need to clarify. First and foremost is that mRNA is read in a 5' 3' direction. Then we know that while it is translating, as I have already told you in the previous lecture also, if you are not aware, you need to see the previous lecture, previous video. The translation of the 5' end represents actually the amino terminal, and so the first codon will always represent the methionine, and which is the amino terminal of the protein being synthesized. So amino terminal methionine will be there, and usually the prokaryotes and eukaryotes contain two different types of tRNAs which can decode AUG. The start codon AUG is recognized by different tRNA, and the AUG, which is in the middle of the mRNA at somewhere, is recognized by some other tRNA. So there are two different tRNAs to recognize the AUG. The initiator codon is recognized by the initiator type of an tRNA, while as the internal AUG is recognized by different tRNA. So these two different kinds of an tRNA are involved in the decoding of the AUG. The initiator AUG codon is actually incorporating the two types of an residues into the polypeptide which is being synthesized. If it is prokaryotic cell, then the initiator codon AUG will code for the N-formyl methionine, and in eukaryotes it incorporates methionine. While as the second kind of an tRNA will recognize the AUG, which is located within the mRNA sequence. Uh, Another aspect which is important for the tRNA, which recognizes the codon through anticodon interaction, is that this tRNA, which decodes for the AUG in prokaryotes, has to incorporate the tRNA FMET, which is called as tRNA FMET, and then the tRNA which is decoding for the AUG in eukaryotes is called as tRNA MET. The difference between them is that one is carrying a formyl methionine attached to it, and another tRNA MET is containing the methionine attached to it. However, both tRNAs are charged by the same enzyme, which is methionine tRNA synthetase. So it means that during the charging process of this tRNA in both prokaryotes and eukaryotes, the charging happens with the help of a methionine, as you can see in the accompanying picture. Then in prokaryotes, since it has to distinguish between the AUG starter codon and internal AUG codon, what happens for this initiator tRNA, which decodes the initiator AUG, there is a secondary modification to the methionine residue. What happens is that the transformylase enzyme methylates the methionine attached to this initiator tRNA to form N-formyl methionine. This is the enzyme which is specialized in it in the transformation, and it utilizes the formyl group from the tetrahydrofolate and 10 formyl THF, which acts as a donor of the formyl group. This formyl group is sort of a acts as a barrier for the degradation of the first methionine residue, or also acts as a tag for it for the start of the polypeptide. So when an initiation has to take place, it requires certain things which are necessary for the initiator complex to form at this initiator codon. The process of initiation for the polypeptide synthesis in bacteria will require these things: the 30S ribosomal subunit first, then it will require mRNA which is to be decoded, then the initiator type of a tRNA which is fmet tRNA, fmet which is charged with the formyl methionine residue, and then specifically certain initiation factors which are initiation factor 1, 2 and 3, and an energy which is given by the GTP moiety, then 50S ribosomal subunit so as to make a full functional ribosomes together with the 30S, and for the stabilization the ions magnesium. So this whole assembly which is there in the initiator is formed in a stepwise manner so that the initiation process will begin and the synthesis of the polypeptide will start. The initiation process in itself is a stepwise process. It involves three different steps. In the first step, the 30S ribosomal subunit will bind to the initiation factors IF1 and IF3. The function of them is to prevent the premature binding of the tRNA to A site, particularly of IF1, and then IF3 will prevent the interaction of 30S and 50S so that they will not combine beforehand before the binding of the tRNA, fmet tRNA. So the mRNA that binds to the 30S subunit is actually directed by the Sheen-Dalgarno sequence. In the second step, what happens is the GTP bound IF2 and the initiator fmet tRNA fmet will bind to the mRNA molecule, and the initiator tRNA will bind to the P site, as I have already cleared in the previous video. So the first initiator tRNA, which is bound to the fmet residue, will bind directly to the P site, peptidyl site, as you can see in the picture here, and not to the A site. That's why, because IF1 is also there, it will prevent the initiator tRNA from binding to the A site. So whatever will come, the first initiator together with the GTP will bind to the P site directly. So the interaction between the anticodon of tRNA and the first codon of the mRNA will happen in this phase. And after this, IF3 will be released. IF3 is released, what happens is that the 50S subunit is able to interact with the 30S subunit and make a complete assembly of the ribosomes, so that the initiation process can now be told that it is completing the initiation process. So in step 3, the whole assembly comes together and all the initiation factors will release, and the GTP, which is bound to the initiator codon, will be hydrolyzed to GDP, and this energy release will allow the interaction to happen between 50S and 30S, making the whole initiator assembly, which includes the mRNA to be read, the tRNA fmet tRNA initiator tRNA, and the binding of 50S with the 30S. So at the end of the initiation phase of the translation, what contains the functional initiation complex is the mRNA, which is to be read, the initiating fmet tRNA fmet at the P site, while as the 30S and 50S subunits are also together. So the correct binding of the initiator fmet tRNA fmet in prokaryotes at the P site results in the completion of the 70S initiation complex, and usually three different points of recognition and attachment are there. First and foremost, I have already told you that this whole assembly is mediated through the interaction by the Sheen-Dalgarno sequence in the mRNA and 16 sRNA of the ribosomes. In the previous lecture, I have shed a light on this. Then there is an codon and anticodon interaction, which means that the tRNA FMET anticodon recognizes the initiator AUG codon, and it is fixed at the AUG at the P site. And then finally, the interactions between the ribosomal P site in itself with the FMET tRNA, FMET. These three enable the correct binding of the initiator tRNA out of all these tRNAs which are involved, and so that it will decode the AUG for the initiator FMET amino terminal of the polypeptide, which is being synthesized from the decoding of mRNA by this machinery. So this will end the initiation phase of the translation, and then the complex will move to elongation phase. Eukaryotes, the initiation of the translation encompasses this similar kind of a mechanism which we just went through. However, there is some difference in the circularization of the mRNA which is to be translated or which is to be read. This mRNA which is starting the translation process is actually interacted by two different complexes of the proteins. One is the poly A binding protein, which binds to the 3' and poly A tail, while as the initiation factors, eukaryotic initiation factor AGE, will interact with the 5' end of this mRNA, which is to be translated. And the interaction of PAB and eukaryotic 4E initiation factor will cause the bridging between the eukaryotic factor G and the eukaryotic factor initiation factor 3. And this interaction is actually mediated through eukaryotic initiation factor 3, because it interacts with the crevice or the nook of the 440S ribosomal subunit of the eukaryotes. So eukaryotic ribosomal subunit in itself is somewhat specialized in having certain crevice which interacts with the initiation factors and then enables the circularization of the whole mRNA molecule on this crevice, so that the not only the mRNA will be bound in a circular manner and will be transcribed and available much more avidly than in the prokaryotes. As also this enables the ribosomes to be seated near the ribonucleophorins which are present on the rough endoplasmic reticulum. That's why the circularization of the mRNA here in eukaryotes is important for the initiation of the translation. This is just the difference between the prokaryotes and the eukaryotes. Rest of the translation initiation is almost similar. Second difference is in the tRNA. The initiator tRNA which binds to the first AUG codon in the eukaryotes will always be tRNA MET tRNA, which contains the charged methionine residue. It is not formulated as in the prokaryotes. Second phase of the translation is the elongation phase. It is essentially same in both bacteria as well as eukaryotes. In this phase, amino acids are brought to the mRNA by the help of the charged tRNA, and one by one they are added based upon the codon anticodon interactions, and then the polypeptide which is being synthesized grows one at a time till the end happens. So the elongation phase, like the initiation phase, also has three steps or three stages: recruitment, transpeptidation, and translocation. Step of the elongation phase is the recruitment process. The recruitment process helps the binding of the second tRNA to the amino acid site. As you can see in the accompanying picture, the incoming tRNA is there with the appropriate amino acid charge to it. It will bind with the help of the elongation factor TU, which is bound to the GTP. The whole complex here will be formed first, and then it will be bind to the initiation complex at the A site to make it a elongation step. So the elongation step will begin only when the second tRNA will be interacting with the codon which is present at the A site. So after binding the GTP, which is here associated with the elongation factor, TU will be hydrolyzed to the GDP. The energy released will help in the proper interaction between the anticodon and codon arm, so that the appropriate amino acid will be incorporated as against the codon which is present in the A site. The hydrolyzed TU elongation factor will then be regenerated back, so that it will do the function again and again. And this process on this step will be the recruitment step of the elongation. At the end of the recruitment, the GTP associated with the EFTU, or in eukaryotes EF1 alpha, is hydrolyzed and then ejected, and EFTU GDP is usually inactive, so it has to be replaced with the GDP, and EFTU can now be reused only when GTP is associated with it. So this finishes the first step of the elongation process. The second step of the elongation process of the translation involves the creation of a peptide bond between the amino acids present in the P site and the A site. What happens actually is the peptide bond, which is between the amino acid and the adenosine of the tRNA, is first broken, and then the another bond is formed between the carboxyl group of the first amino acid and the amino terminal of the second amino acid, as you can see in the picture. The initiator tRNA is then decharged. It is devoid of any amino acid, and then transpeptidation, this reaction of the creation of bond between the two amino acids, is the function of the large subunit of the ribosome, which is having a peptidyl transferase activity. So this is how the bond is formed between the first amino acid and the second amino acid of the tRNA. The amino group of the second amino acid creates a nucleophilic attack on the carboxyl end of the first amino acid, which is methionine, for the creation of this bond. This is mediated by this large subunit of the ribosomes through its peptidyl transfer. Then the third step of the elongation is the translocation, which involves the movement of the whole subunit, one codon, ahead of the mRNA towards the 3' end. And this also involves that the initiator tRNA, which is now decharged, will be exited from the complex. In terms of the prokaryotes, in bacterial cells, there is a E site, so that this decharged tRNA will move to the that site. However, in the eukaryotes, the tRNA which is decharged of the amino acid will be moved out of the complex. The methanoyl residue is thus tethered to the amino acid, which occupies now the A site, and then the tRNA in the A site is now transferred to a peptidyl tRNA. That means that now this whole complex will move one codon ahead towards the 3' and this will mark the translocation process of the elongation. Phase of translation is the termination process. Termination means that now we have synthesized the whole peptide out of the information by decoding the mRNA, and now it is to be stopped. The termination codon present in the A site, when it happens, the termination codon, as you know, there are 3 different termination codons. When they accompany or occupy the A site, then the termination has to begin. But we know that there are no tRNAs which recognize the termination codons. So what happens is actually there are certain factors which bring out the termination. Those are called as the release factors. In prokaryotes, the release factors are of 3 different types: RF1, RF2, and RF3. RF1 recognizes the 2 stop codons, and RF2 recognizes another 2 stop codons. So one is common, that is UAA. UAE is recognized by both RF1 and RF2. Then RF3 is important in assisting the function of RF1 and RF2. In case of eukaryotes, the release factor is the eukaryotic release factor. Only one kind of a release factor is present there, which brings out the termination process. Actual process of termination is dependent upon initially by the release factors. The release factors are able to recognize the stop codons at the amino acyl site. They are recruited to it, bind to it, and get activated. After activation, the release factors catalyze the transfer of the polypeptide, which was actually bound to the tRNA at the P site. This polypeptide is then transferred directly to the water molecule, and as soon as it happens, it is released from the whole complex. However, the complex which encompasses the tRNA, mRNA, and the two subunits of the ribosomes are not able to disassociate with the help of release factor only. There are two other factors which are needed for the disassociation of this complex and the release of the uncharged tRNA also from the P site. This is done with the help of another two factors, which are ribosome release factor RRF and ribosome elongation factor G. Elongation factor G is important in instilling the GTPase activity. This feeds the energy for the disassociation of the whole complex of this translation machinery by interacting with the release factors. So at the end, when this disassociation happens, immediately after the disassociation, the elongation factor G activity, which releases the energy, recruits the initiation factor 3 also to this specifically to the small subunit. And we know that initiation factor 3 is critical for 30s subunit, so that it will not interact with the 50s subunit and bind without the availability of the initiator tRNA. So I have three binding encompasses the termination process. Termination is not until and unless the 30 subunit binds to the initiation factor 3. So with this lecture, we completed the translation process. In the next lecture, we will be discussing the inhibitors of translation and some other tidbits about the translation. If you like the contents of the video, you can order the book Molecular Biology and Biotechniques from Amazon. It's available both in Kindle and paperback format. And do subscribe to my channel for further videos which I will upload in due time.